much, Almud, for the introduction. And I've shortened my title because I'm very excited to share our team's effort on developing this single cell imaging and control of microbial metabolism via the structures which we call protein nanowires to mitigate three of the biggest challenges of our generation: pollution, climate, and diseases. So every living cell needs to get rid of electrons when nutrients converts into energy, and this is the reason why we are breathing now, so that the excess electrons can be taken by oxygen and that forms water, and this all happens inside the cell. However, most life on our planet has to figure out how to survive without oxygen, and many bacteria which have been evolved way before the great oxidation event, they respire by a process called extracellular electron transfer, or EET for short, where this hair-like filament, which we call nanowires because they work as a highly efficient make to transfer electron. And in a natural environment, they transfer electron to minerals in the soil such as iron. And we can replace that mineral with an electrode, and these bacteria have been used to generate electricity in a continuous way, and that has driven many bioenergy and biofuel and bioelectronics applications. The reason why these nanowires are globally important because they can enable microbes to survive in extreme environments such as very hot or a very cold temperature, at a very deep pressure, very acidic or a basic condition, or even the radiation 100 times lethal to humans. These nanowires have also enabled microbes to perform useful tasks like bioremediation, so they can clean up oil spills or the contamination of heavy metals such as uranium. So although these nanowires are known to be important to mitigate these challenges of climate change, pollution, and diseases, the nanowires' properties and their composition was unknown despite decades of research. And this is why I'm very grateful to Almud and HFSP for supporting this work, which have allowed us understanding of this nanowire, which helped us to demonstrate that these nanowires enable this extracellular electron transfer at unprecedented rates and distances. So here I'm showing an electron microscopy image of a bacteria called Geobacter. Geo is Earth, and it's one of the most common bacteria on our planet. And those hair-like filaments, I've colored them in red. Those are the ones which act as a nanowire, and they basically allow these bacteria to export electrons outside their body and transfer it to extracellular acceptors or other bacteria. And what we found that these electrons can move at ultra fast rates within 100 femtosecond and across distances of centimeters, which is 10,000 times the size of a microbe. So you can think of these nanowires as a snorkel, but that's 10,000 times your size. So in a human term, it's 10 kilometers long for a meter long person. So this is a remarkable feat this bacteria achieve. So the technique we use to identify the nanowire composition is a cryo-electron microscopy, and that showed that these nanowires are made up of polymers of cytochrome. So they have a heme just like hemoglobin in our body, but here the heme forms a linear chain, and then that protein can polymerize into a wire with all hemes very closely stacked within each other. So they just look like our phone charger. So here I'm showing you every unit in a different color with corresponding hemes in the same color, and the hemes form the wire to transfer electron, and the protein wraps around it like an insulation. And we and others have found many different nanowires. They are called outer membrane cytochromes, or OmcS, S, G, and E. They all have very different properties, different conductivity, different way they assemble, but they all have this common heme chain at their core. We have just started through HFSP support, try to understand why most cytochromes cannot polymerize and what is so unique about these cytochromes that they form this wire. And we have been able to identify a distinct gene cluster which drives the assembly of this wire. And we have also been able to identify the function of key proteins which are necessary to assemble these wires. And what's really exciting for us, which came out of this HFSP support, is these nanowires are not limited just to few species. They are much more widespread in a diverse species and environment. For example, in addition to geobacter in the red color, many archaea, which are the most ancient ancestor of us, they also show many of these nanowire genes highly expressed, and many of them are important for our climate, such as breaking down methane. And many bacteria, which I showed in the beginning, which grow at very high temperatures and pressures, they also show this OmcS nanowire gene cluster. And remarkably, the bacteria or archaea make only one kind of wire, so they don't make multiple wires. So bacteria primarily make this wire called OmcS, whereas archaea exclusively make a wire called OmcZ, and we are trying to understand how do they decide which wire to make. So far, the studies had been limited to sheared cytochrome. So we separate those from bacteria, like giving them a haircut, and collect those hairs, which are this filament, and measure their properties in vitro. And one of the things we were able to do with HFSP support is develop the in vivo imaging of this electron transfer to answer this central question that do bacteria are actively using these nanowires during their metabolism for this extracellular electron transfer in vivo. And this technique is called electrostatic force microscopy, or EFM, and this can basically measure the electric field, and from that you can image the electrons. So to do that, what we do is we grow this bacteria on an electrode called graphene oxide, which itself is a non-conducting, but when bacteria export those metabolically generated electrons to the graphene, it becomes reduced graphene oxide, or RGO, which is electronically conducting, and this technique can separate between conducting and a non-conducting region, and this allows us to exactly map which wire is used by bacteria at what efficiency. So first we use atomic force microscope to image the bacteria and ensure that only one kind of wire is produced by them, and then we image them before any electron transfer and a conductivity, and we compare that image after the electron transfer. And the white color you are seeing are actually the electrons metabolically generated by this bacteria and transferred to the graphene, and that's why that region has become conducting. And this method has proved very useful for us because we can now compare how bacteria use different kind of wire. One of the biggest question in the field was, what is the wire? Are they using a PilA filament? PilA in Latin means hair, and it was thought for 20 years that these wires are made up of PilA protein. And using this technique, we were able to show that in the presence of PilA, the bacteria are not able to export electrons outside, and when you have the most conductive nanowire like OmcZ and OmcS, the bacteria can do a very fast electron transfer over a long distance. So we have been using this now to really control the growth of the bacteria. So we have been able to use diverse physiochemical stimuli, which enhances the conductivity of this nanowire, and that leads to acceleration of bacterial growth. So we are really excited that now we have a tool to not just understand the mechanism of how electrons are moving, but use that mechanism to control this bacterial growth. So our team has what really HFSP enabled us to bring people from different parts of the world with a very different expertise. So we have been able to team with experts in this PilA protein, the system which secrete protein, and my colleague Carlos, who is in the audience, who is a world expert in the cytochrome protein, and to understand how the nanowires connect to intracellular protein. So I'm going to start with the work led by Carlos to answer this key question about what connects bacteria to nanowires. So it's not sufficient to have a wire on the surface. The cells still need to have a way to actually connect electrons from inside to those outside wires. So what are those connecting plugs which are allowing this wire? And this is one of the key questions Carlos has helped us to answer. And what we realize, just like biology, there is never a unique answer, and depending on the environment, the bacteria use different plugs. So the first plug we found they use is a protein complex called OmcC, and they use that plug just to get enough energy so that they can kickstart synthesis of nanowires. and then they can use periplasmic cytochromes, which Carlos is world expert in, to demonstrate that they can transfer electron to ion. And finally, we also discovered in some cases the bacteria even make wires inside themselves in a periplasm, and that allows them to grow on the electrode. So I'm just going to show you the cartoon here, and I urge you to stop by our posters for the details about this work, where Carlos has used this beautiful NMR spectroscopy to see how the proteins are binding and transferring electrons to each other. So what we found is, in the beginning, bacteria don't need nanowires because they can just get started with whatever is on the surface. And in that case, they use this protein complex as their initial connecting plug to get electrons out, and that gives them the initial energy, which allows them to assemble nanowire. And then Carlos's group was able to show that at that stage, this periplasmic cytochrome called PPC can bind to this wire and directly transfer electron. And this then allows this bacteria to transfer electron to minerals such as iron oxide. And in the case where these bacteria are generating electricity on an electrode, what we found is they make wires inside them called EXTA, and that allows them to transfer electrons much more rapidly. And then they use another plug and a wire called OmcZ to transfer electron, and this is what allows them to generate electricity by transferring electrons to the electrode. So the reason why this work was so surprising, and the reason I'm so grateful to HFSP, because this work really challenged a 20-year-old notion that the nanowires are made up of this PilA protein. And there were thousands of papers believing that these nanowires are made up of this protein called PilA-N. And most of the data was genetics, where if you delete the PilA-N gene, you don't see bacterial growth or you don't see the filament. So the question is, now we have identified that these nanowires are polymers of cytochrome. We had to answer this question, that what is the role of this PilA-N protein in the formation of this nanowire. So again, we use a technique, cryo-electron microscopy, and we were able to show that this bacteria form a bipartite, like two parts of a PilA protein, and which structure and property is inconsistent with nanowire. So here I'm showing you a cryo-electron microscopy structure. And although for 20 years it was thought that the PilA-N, which is shown in orange, is the nanowire, we found that that protein cannot assemble by itself. It needs a separate protein called PilA-C, and they come together and form a filament. And if you look at just the PilA-N protein, it's very, very hydrophobic, so it cannot function as a wire because it cannot even assemble. It needs this completely different protein called PilA-C to protect that part, and that's the hydrophilic part which allows them to come together. And this was very surprising because PilA-N was thought to be the nanowire for 20 years in thousands of publications. So then what's the question of what is the role of PilA if they are not acting as a wire? And we found they have a completely new role. They are required for the secretion of this cytochrome. So if you delete these PilA genes, the cytochrome remains stuck in the periplasm, which I'm writing here in the PP. And when you restore the gene, then the nanowires can come out. And we can also confirm this with the imaging, that the primary role of this PilA is secretion of this protein, which was a very surprising finding. So and other nanowires show also similar result. So through HFSP support, we were able to answer this key question about how do a PilA-like protein can act as a secretion of nanowire. And this is the model we have been able to come up with the results we have obtained so far, where these PilA are acting as a piston, and they push these cytochromes out, which I'm showing here in the red color. And then cytochromes can self-polymerize into a filament. And this system is a very novel system called a hybrid system, made up of two distinct systems, type 4 PilA and a type 2 secretion system. So we were able to demonstrate that the cap protein of this PilA is very critical. So if you delete the cap protein, the bacteria cannot grow efficiently, and they cannot secrete this OmcZ nanowire. And through HFSP support, now we are identifying the mechanism of how this cap has been used by the wire and how it's binding to another protein. And this work is really very challenging because there is no known structure of this cap protein for any bacteria. So as you see, the structure is very complex. It has many different parts, and this really challenging problem has been taken by Lisa Craig, who is a world expert in type 4 PilA, and her lab has been working very hard to understand the structure of this tip, and they have been using multiple strains to both express and purify different parts of this tip protein to understand, how do they look like and how do they interact with the protein. So the next question is, what is the machinery which allows these nanowires to assemble and secrete? And there are two main known machineries in bacteria. One is called this type 4 PilA or a T4P, and the second is a type 2 secretion system or T2SS, and they both work very differently. Type 4 PilA are expressed on the surface, whereas the type 2 secretion system PilA remain inside the bacteria and they secrete the protein. And the answer we found was very surprising. It was neither of those two machines. It was an entirely new machine. And again, this was the work supported by HFSP through a work led by Olivera Francetti group, and she used a technique called bacterial to hybrid assay, and her group was able to show that both some components of type 2 secretion system and some component of type 4 PilA, they both are required in the assembly, and that was very surprising because there was no precedence for such a system, and we were able to confirm that subsequently through genetic studies, and we were able to show that both type 2 and type 2 components are required, which leads to this very surprising model that this is a new kind of machine which is using some part from type 4 and some part type 2. To evaluate if this model is correct, we use a technique called electron cryo-tomography in collaboration with Jun Lu at Yale, and their lab member Shoichi was able to identify the machine of this wire and build its structure by averaging it and understand the symmetry. And the structure also confirmed that this is indeed a unique machine, never before been observed, where it uses several parts from the type 2 secretion system protein and several parts from a type 4 PilA protein. And if you look at the evolution of this wire, which I'm showing you here in the red color, even the evolution shows that this assembly machinery is independently evolved from all other machineries. So they are not part of either type 4 or a type 2. They have their independent line of descent. And another thing came out of this research is this machine is not limited just to Geobacter group of bacteria, but it's also present in diverse species and environment that need this extracellular electron transfer. So finally, we have a clue on how these bacteria assemble this wire, and that allows them to basically respire without oxygen. So what Olivera Francetti group is now working on is reconstitute this machine to understand how these nanowires are assembled, and the way they are doing it is expressing both type 4 and type 2 system. And I'm running out of time, so I'm just going to summarize that they have been able to clone this entire gene cluster in E. coli and identify different parts of this machine, and the same they are doing with the type 2. And this is a really critical work to understand how this machine works. And with that, I want to end at one of my favorite quote is, life is nothing but an electron looking for a place to rest. And really, these microbes are using electrons to, respire without oxygen, and that led to this really simple model. How can they use this different wire? And our hope in the future is we can now use this bacteria as a factory using this very fast speed, because these materials can be flexible, they can regenerate themselves. So there are several applications we and others are looking into, which came out of this basic mechanistic studies. And with that, I want to thank all of you and HFSP for their support, and please stop by our posters. Thank you.